What if an AI could unlock the secrets of life that had stumped scientists for half a century? Now, imagine that same AI just won a Nobel Prize. We're talking about AlphaFold and its co-creators, John Jumper and Demis Hassabis, who just received the Nobel Prize in Chemistry for fundamentally transforming our understanding of proteins. For Hassabis, this wasn't just a project. It was the driving force behind his entire career in AI, the very reason he founded DeepMind. It was exactly 5 years ago this week that AlphaFold 2 burst onto the scene, stunning the scientific community. The initial hype has settled, but the impact? That's what we really want to explore. How are scientists using this groundbreaking tool, and what's truly next for this revolutionary technology? When I spoke with John Jumper, he shared, with a laugh, that it's been an extraordinary 5 years, adding that it's hard to remember a time before I knew tremendous numbers of journalists. And that's a pretty good indicator of the magnitude of their achievement. AlphaFold has rapidly evolved since its debut. We've seen AlphaFold Multimer, which can predict structures involving multiple proteins working together, and then AlphaFold 3, the fastest version yet. DeepMind didn't stop there. They unleashed AlphaFold onto UniProt, a massive protein database used by millions of researchers globally. Today, AlphaFold has predicted the structures of an astonishing 200 million proteins, essentially, almost every protein known to science. Despite this monumental success, Jumper remains grounded. He reminds us, that doesn't mean that we're certain of everything in there. It's a database of predictions, and it comes with all the caveats of predictions. This humility is refreshing, especially considering the problem AlphaFold tackles is incredibly difficult. Think of proteins as the tireless, microscopic machines that make all living things work. They form our muscles, grow our horns and feathers, carry oxygen throughout our bodies, ferry crucial messages between cells, fire our neurons, digest our food, and power our immune systems. Understanding exactly what a protein does and its potential role in diseases or new treatments absolutely relies on figuring out its three-dimensional structure. And here's where the challenge lies. Proteins are long, intricate strings of amino acids. Chemical forces then twist these strings into incredibly complex, specific knots. Imagine trying to deduce the exact final shape of a complex origami animal just from a flat piece of paper and a random list of folds, without knowing the precise order or angles. An untwisted amino acid string offers very few clues about the structure it will eventually form. In theory, most proteins could fold into an astronomical number of possible shapes. The core task is always to predict the single correct one. Jumper and his team built AlphaFold 2 using a type of neural network called a transformer. If that sounds familiar, it's because it's the same underlying technology that powers large language models like ChatGPT. Transformers excel at paying attention to specific, critical parts of a larger, complex puzzle, making them perfect for untangling the elaborate folds of a protein. So here we are, 5 years after a groundbreaking AI changed biology forever, and the people behind it have a Nobel Prize to show for it. We're witnessing a fusion of artificial intelligence and fundamental biological research that isn't just accelerating discovery, it's rewriting the very playbook. The implications for medicine, biotechnology, and our understanding of life itself are almost unfathomable. What new frontiers will we unlock next, now that we can essentially read the blueprint of life with unprecedented clarity? If you found this dive into AlphaFold fascinating, be sure to hit that subscribe button wherever you get your podcasts, and let us know what you think about AI's role in scientific discovery.